Okay, thank you, Mario, for the introduction. My name is Zichen Wang, and I'm a PhD student in Dr. Avi Mayan's lab. So today I'd like to present to all of you about my project that use the links L1000 gene expression data together with some other features of drugs to predict adverse events. So I'll first start with some brief background. So I think everybody here should be very familiar with the Links L1000 project performed by the Broad Institute. So basically they are trying to make this data cubes where one of the dimension is different types of perturbations, and the other dimension is to perturb different human cell lines, and then use different phenotypic assays to gather the data from multiple aspects. So in this project, we are particularly interested in chemical perturbations. So in the Links L1000, they profiled over 20,000 drugs or small molecule compounds in different human cell lines, and then they and then after different periods of time, they use this L1000 technology to profile the gene expression of those human cell lines, and then generate a lot of data. And also there are other similar efforts that also generated a lot of data about drug or small molecule compounds perturbations within human cell lines. For example, this one, Molecular Library Probe Production Center Network, has used this multiplex cytological assay to profile the morphological change of human cell lines after the treatment of drug compounds. So here is a brief workflow of this project. So basically they treat the cell with drugs or small molecule compounds, and then they did this cell staining assay where they are able to stain different organelles within the cells, and then they used image capture and automated image analysis to extract a set of morphological features within the cell that are representative of the morphological signatures of drugs and small molecule compounds. And so since we have all of those large-scale projects and generated a lot of data about both drugs and small molecule compounds, we want to study the effects of drugs. And in my project, we are particularly interested in studying the adverse drug reactions, and here's the background of ADRs. So ADR is an appreciably harmful or unpleasant reaction after taking a drug. And then there's a very closely related term, side effect, which is a therapeutic or adverse secondary effect also from taking a drug. So the difference between ADR and side effect is that side effect can also be beneficial or therapeutic, whereas ADRs are usually bad or adverse reactions. And it's very important to study ADRs because it's a major concern of the public health, and also it's very important during the drug development process. And if you fail to identify severe ADRs, it will lead to patient morbidity and the withdrawal of drug from the market. However, from the scientific point of view, ADRs actually contain rich information about human phenotypes. So here is a graph showing the different entities that are interested in by the system biologist, and we can see that side effects is closely related to drugs as well as mouse phenotype and diseases. So a good knowledge of the mechanisms of side effects is pretty important for studying the whole system. So there have been many previous efforts that people have done to predict ADRs, and initially people used chemical fragment-based approaches to study ADRs, and those models are based on the quantitative structure activity relationship, or QSAR models, and these models assume that the biological activity of small molecule compounds can be quantitatively expressed by the chemical properties of the compound. And more recently, people started to integrate biological features into those chemical-based models, and people have also shown that the integration of biological features can improve the predictability of ADRs. However, most of the studies listed here are the biological features that are used in those studies are based on the target proteins of those small molecule compounds or drugs, and there is a limitations for that. So for a drug, you may have multiple targets and maybe off-target proteins that have not been identified, and there are studies have shown that the off-targets of drugs sometimes can explain better for the mechanisms of ADRs. And also focusing on target proteins, those models cannot be applied to potentially investigational drugs that don't have target proteins identified. So that could be a problem of those type of models. And in this study, we are trying to collect multiple drug attribute tables from both phenotypes, biological and chemical point of views. And then after we have those attribute tables for drugs, we can train predictive machine learning models to predict those ADRs, and we can also potentially extract knowledge from those predictive models. And we also want to apply our predictive models to all of the small molecule compounds profiled in the LINKS project and share the predictions through a web portal. And also we have been trying to set up crowdsourcing challenges to challenge people to come up with their predictive models, and we can potentially evaluate their predictive models. So I'll start with the first part, which is the collection of the drug attribute tables. So to study the ADR, we need a good terminology for identifying those ADRs, and thankfully there is a medical dictionary for regulatory activities, or MEDRA here, and this dictionary is internationally used by clinicians and regulatory agencies to identify adverse drug reactions. And it has 5 different hierarchies here, from the highest to the lowest, and those different hierarchies of terms are connected based on anatomy, pathology, physiology, or etiology of those adverse reactions. And the levels of terms that are used to directly describing side effects or ADRs are preferred terms, like this level, and also you can trace those terms up to their upper levels. So also we need to collect the gene expression data for drug compounds to as their attribute tables. So here the goal is to collect a gene expression signature for each drugs or small molecule compounds from the LINX L1000 project. And as we know, there are different levels of data in the LINX L1000, and there are also different types, different ways to get the gene expression signature from these data. And so we can directly get the signatures, which is modulated z-scores computed by the root. And we can also use another way, which is to use characteristic directions, a method developed in our lab to compute a differential expression signature for each set of drug perturbation experiments. And then we were able to show that our method is slightly more predictive of ADRs compared to the z-score. And also from the MLPCN project, we can get morphological signature for each drugs. And in this data set, they have already processed the imaging data, and we can directly get the morphological descriptors based on their analysis. And finally, we collected the chemical structures of all the drugs and small molecule compounds in those projects. And to convert those chemical structures to chemical descriptors, there are many structural fingerprints that can be used. And for this project, we simply use the 2D structural fingerprints called MAX, which has 166 structural keys in the bit position. And here is basically a figure showing how you can, convert a drug structure to a fingerprint, to a chemical fingerprint. And also, and the next step to do is to map between those different projects, map the compounds used in those different projects to FDA-approved drugs recorded in the side effect resources called CIDR. So the way we mapped those drugs was by their chemical structure encoded by the smell streams, and following those procedures. So as a result, we are able to map around 800 chemical compounds profiled in the LINX or the MLPCN to the FDA-approved drugs in CIDR. And here is basically a Venn diagram showing this overlap. And here is a simple graphical abstract for making the predictive models. So we have the shared drugs on the rows, and we have those three matrices representing the gene expression from the L1000, the chemical structures using the MAX keys, and the cell morphological features from the other project. And then we can use either a single of those matrices or the combinations of any possible combinations of those matrices to predict side effects matrix here inside there at different levels of terms. For example, we can predict them in the lowest level term or preferred terms, as well as higher level grouping terms. And the machine learning models we used here is called multi-label classification. And I'll give a little bit of background about this multi-label classification. So multi-label classification is a variant of single-label classification. So in a single-label classification, the goal is to learn a function f from the matrix of features, whereas the rows are each are instances, and the function f take a vector of features and then output a single number, binary number, or a possibility between 0 and 1, representing the membership of whether this instance belongs to this label. And for multi-label classification, the goal is to the goal for this function f is to output a vector of labels representing the membership between the instance and the list of labels here. So there are multiple methods for this multi-label classification. The baseline approach for this type of task is called binary relevance, which is to train a classifier for each label and ignoring the correlations between labels. So for this stage, I primarily used the binary relevance for this learning task, and also for multi-label classification, there are different types of evaluation metrics for evaluating for benchmarking the models. So you can either do label-based evaluation or instance-based evaluation. So label-based evaluation, in my case, is to evaluate to see how well my model is doing in assigning the right drugs for each side effect. And for instance-based models, that is to look at how good the classifiers is doing in assigning the right side effects for each drug. So then I started to make those machine learning models in threefold cross-validation schemes. So all of the evaluation plots shown here are based on the average of threefold cross-validation. So here I basically only used the 251 drugs that have both gene expression profiled in L1000 and morphological features profiled in the other project. So we can see that the blue is the performance of gene expression in predicting ADRs at the preferred terms level. So you can see for the instance-based variations, gene expression is the most predictive among the three single matrices, and chemical structure, followed by chemical structure, and then morphological features are not very predictive. And then we can also see that when we combine gene expression with max, with the chemical structure, we get even better performance, and that have also been proved by multiple previous studies. And So when looking at label-based evaluations, which is basically to look at for each side effects or ADRs, how well are we doing in predicting each single of those. And we can see that gene expression still is the best predictor for most of the ADRs, followed by chemical structures. And combining gene expression with chemical structures can still significantly improve the predictability for each ADRs, or for those out of those ADRs as a whole. And I tested those two with the Wilcoxon rank sum test, and it is significant that this one is significantly larger than this one. So so we can see here the morphological features are not very predictive of ADRs. So then, based on this Venn diagram, we decided to drop the morphological data and then only use the gene expression and chemical structures, so that we get more drugs in the to when training the predictive models. And then we looked at where those gene expression signatures came from for those drugs. So here I have those pie charts showing the portions of those gene expressions that are stems from. So we can see most of the gene expression signatures were generated from the MCF7 or PP3 cell lines, and when categorizing those cell lines into the types of cell lines, we can see most of those are cancer cell lines, and also a small portion of those are immortalized normal cells or primary cells. And also, and then when we look at the dosage, we can see the majority of those signatures were derived from treating the cell with 10 macromillar of drugs, and then after 6 hours, and and small parts are derived from after 24 hours. Then we used the drugs that are shared in the links in the FDA-approved drugs, and then keep making the predictive models. And we can see the gene expression is also still very predictive when expanding the instances, the number of instances, and as well as chemical structures. And here we can also basically show the same thing, that gene expression plus chemical structure can be more predictive of ADRs. And then we want to further improve our predictability of ADRs by converting the gene expression signatures to the enrichment vectors of gene ontology terms, that is to transform the gene expression data to see what gene ontology terms are enriched, and then use the enrichment vectors of gene ontology terms to predict ADRs. And we can see this brown color here is stands for the goal terms here, and then goal term is more predictive compared to gene expression, and the combination of goal terms and chemical structures is a little bit more predictive here. And the same is for the label-based evaluations. When we show those results to other people. The commonly asked question was that how good your prediction is doing compared to other published methods. But the problem is that there is only a few studies that have reported their evaluation statistics, and also there is only one data set that have recently came out for people to benchmark the predictive of ADRs, and then this data set here is called the OMOP, and then this data set only contains 4 ADRs and a small set of drugs. So I chose a very recently published paper, and then they trained their model and cross-validate their model with this small data set, and that's their performance. So I also get this data set and I use my predictive models to on this data set, and you can see that my predictability for those 4 types of ADRs are better than what they published. So then back to my other goal is to interpret the performance of those classifiers to potentially get some knowledge about the mechanisms of ADRs. So I have shown that the goal terms are very predictive of many ADRs, and then by interpreting those predictive models, I can associate goal terms with ADRs that they are very predictive of, and then I was able to make this network of ADR and goal terms, and in this network ADRs are in triangle and goal terms are those circles, and in this network we can recover some known ADR or ADR goal term connections, and here are a few examples. So you can see anemia here is associated with heme transport and heme transporter activities. Those are goal terms, and apparently anemia is a condition where the body does not have enough healthy white blood cells, and as a result, the heme transport, the heme is very important for a very important component for the red blood cells, and as a result, those terms are connected with each other. And there are a few more examples. I think I'll just skip them, and right. So here there's another side effect or ADR that is called cardiac fibrillation, and my predictive model suggests that those 2 goal terms, that are those 2 axon or neuron projection related goal terms, are very predictive of this cardiac fibrillation. And then I looked at the PubMed and found a paper published very recently describing the potential connection between the cardiac fibrillation and neuron projection. And also there are more examples. When I did the enrichment vector methods, when I use the enrichment vector methods to transform the gene expression data, I also tried various of different gene set libraries that contains different aspects of biological terms. And as we can see here, there are also some other gene set libraries that are also very predictive of ADRs. For example, the CCLE, which contains the signatures of differentially expressed genes in cancer cell lines, are very predictive of ADRs, as well as this one, Uber, which is a cross-species phenotype ontology. Sorry? Hello? Hello? Did anyone ask any questions? Hello? Can anyone hear me?
Yeah, we can hear you. I think it was a mistake. You should continue.
Oh, okay. Okay. So I will just proceed. So this Uber is a cross-phenotype ontology including human phenotype, mouse, and zebrafish phenotypes, and then they associate genes that are when knocking out have exhibit those phenotypes with those phenotypes. And then we find that when we enrich the gene expression signatures to these phenotypes, ontologies, this vector of phenotypes are also very predictive of ADRs. And then we can perform a similar analysis as we did for the Go terms. And this time we can associate ADRs with those phenotypes. And the good thing about doing this is that we have a ground truth ADR mouse phenotype connections that were manually collected by a pharmacologist. And then we can benchmark the network of the ADR phenotype connections based on this. And then in this network, we are able to recover those 4 connections. For example, here, the pigmentation disorder side effects is can be predicted by those 3 mouse or human phenotypes that are related to pigmentation. And also here, leukopenia is can be predicted by the mouse phenotype here, the absence of CD4 positive alpha beta T cells. And then we are still trying to figure out the genes that are responsible for the enrichment of those phenotypes, and potentially we can explain from the gene expression signature data to see what types of genes, when some types of genes are differentially expressed, these drugs can potentially cause those adverse drug reactions. We also applied our predictive models to all the links compounds and links small molecule compounds and drugs, and then we made a web portal to share those results. Here I have this URL for this website, but this website is still under development and the data there is not the newest predictions. but basically we have the prototypes there, and this website can also cross-referencing to other resources, including the side effect resources CIDR, and as well as Links Cloud, PubChem, and Links Information Framework for Life. And then, as I said, we planned to make some crowdsourcing challenge, and here is basically a website hosting the crowdsourcing mega task for the prediction of adverse drug reactions given the gene expression data. However, we didn't get too many impressive submissions, so we didn't include those predictive models in our final predictions. And here I think I'm finished for the presentation for this part, and I would like to thank every members in Dr. Avi Mayan's lab, and I'm ready to take any questions from the audience. Thank you.
Thank you, Zichen. So if somebody wants to ask the question, I will try to unmute everybody, but please unmute yourself if you would like to ask a question as well. All right, so everybody should be unmuted now.
Hello.
Hello.
Hi, this is Jian Bai. A very nice presentation, Jichen. I am wondering, you are very ambitious and you cover so many adverse reactions. Just wondering, because you have done so much, and it's very tough for me to learn because I'm trying to catch up. And you have so many layers. So in your you have gene expression, you have chemical structure. Can you educate me in your chemical structures? How do you get those structural signature or descriptors? How many of them?
Okay. Thanks for your question. So for the chemical structure, we got the chemical structures encoded in SMILE strings, which is a 2D representation of chemical structure for the drugs or small molecule compounds, and we got those SMILE strings directly from Links Cloud, which they provide the metadata for the drugs or compound they use to profile these. And then here is basically a slide showing how you can convert the chemical structures to the structural fingerprints. So for the 2D structural fingerprints, you have basically a list of substructures here, and then for these compounds, you look for those substructures to see whether this compound has those substructures here. For example, these compounds do not have a 4-member ring, but it does have a 7-member ring here, so that you get a bit on the number 19th here, and then you do the same thing for all the substructures defined in the 2D structural fingerprint systems, and then you can get a list of fingerprints that has those key positions. And then for the classification purpose, I converted these fingerprints to binary vectors, which is 0 or 1. So for this, the binary the binary vector has a 1 on the 19th position, the 78th position, and so on, and other positions are 0. So then I use this vector.
Actually, my question is that because you have 2D, you have 166 structural
Yeah.
keys. In
Yes.
your, let's say 881 structural keys, so you almost have 1,000, right? Then you have the 3D fingerprints.
Oh.
And so when you combine all together, are you over? Do we have over 1,000?
Oh, I didn't actually use all of these. So my chemical structure, my chemical features is only the 166 max fingerprints. So the vector here is only 166. I didn't combine them all.
Oh, okay, okay. Then for the gene expressions, you have one slide, and I was going to ask you a question, but I guess there was one slide showed that you combine gene and chemical structure together.
Yeah.
Are we risking overfitting? And then you have the ADR. That's why I was very confused. You have so many ADRs. How do you choose those ADRs?
Okay. So the way I'm combining gene expression and chemical structure is by feature selection, which and the feature selection method that I used is called stability selection, which is a multivariate approach based on logistic regression, and all of those results were based on cross-validation, where I split the training and testing sets. So in theory, there shouldn't be a problem of overfitting. And then, sorry, what is your second part of the question again?
I would normally assume that, okay, you have certain liver toxicity. I mean, you have specific phenotype, and because liver toxicity in terms of clinical manifestation, there are so many different types, right? Like you can focus on fatty liver. How do you, in your so many chemicals, do you have a positive, negative control? And then also when you do the training set, you could have, in your training set, how did you define your training set, and how did you define those ADR? That's why I was. I believe it's my personal preference that it's very important to look at specific clinical phenotype.
Yeah.
Of each organ, right? You are combining across all organs, or?
No. Actually, those drug side effect connections are all from the CIDR database. So, so all
right,
of
but then when I say organs, like you have heart, livers, right?
Yeah.
And you have brain versus, so you have individual organs that adversely affected by drugs, off-target effect or undesired pharmacological target and off-target organ. And so that's why I'm asking that in your ADR, is that you lump all the different across organs and put them together.
Yes.
Bear in mind, there's an organ-organ interaction, so you could have an adverse reaction in the liver, and then that actually affects other part of the organ, other organs. So I think when we use this type of approach, it's very good. But you may have a lot of noise in there.
Yeah. So all these side effect drug connections are from the CIDR database, which they might be drug package inserts. And yes, I think there are many types of ADRs in the CIDR, and I didn't explicitly include or exclude any types of ADRs. I just include ADRs that have frequency larger than 10 to predict. But you can see from those plots that for individual ADRs, they are not equally well predicted or not well predicted. So we can look into what are these predictable ADRs and what are those ADRs that are poorly predictable to maybe learn something about the mechanisms or the organ classifications of those ADRs. Not sure if I addressed your question.
Yeah, I think I would say your work is very impressive, and it's just from my experience, I just give you some comment, and hopefully as your research moves along, then you can refine your research.
Okay. Okay. Thank you.
Zaichen, can I ask you something?
Yeah, sure.
Or just maybe if you could elaborate a bit more on your cross-validation approach, how exactly cross-validated, and then how did you construct ROC curves from there?
Okay, so I think I can start with this slide. Since I use the baseline approach for multi-label classification, so I've first look at each single side effects at a time, and then first I split the drugs to training and validation sets with threefold cross-validation, so 2/3 for training, 1/3 for testing, and then look at each individual side effect, and then use those matrices to train the model for each individual side effect, and then evaluate on the testing sets. And for the raw curves, So for the raw curve here, this is instance-based evaluation. So that means during my training fold, I predict the ADRs for all the drugs in the training set, and then look at each individual drug, and then sort the side effects based on the probability from the classifier, and then plot a raw curve for this one drug. And then since there is 1/3 of the whole data sets of that many drug, so I then use the micro average of those. I then use the macro averaging of those individual raw curves for each drug to generate this raw curve here. And for the label-based evaluation, I didn't actually plot the raw curves here. I just got the AUC for each label, and the label-based raw curves were plotted based on also getting the probability for a drug, let's say a drug X, we get a vector of probability based on the side effects that I predicted, and then sorted the side effects by the probability, and then validate that against the actual side effects that drug X caused, and then plot a raw curve for this this should be for each side effect. So I have a list of drugs, and then I can sort those drugs based on the probability derived from the classifier, and then plot a raw curve for this the side effects and then get the AUC values here. And then I do that for all of the side effects so that I get a distribution of the AUC. And that is the label-based evaluation. Did that answer your question?
Yes, thank you.
Okay.
I have another question. So how many ADRs are we talking about here?
Well, that so for this particular result, since I only use those 251 drugs that have both gene expression and morphological features, and I think here I have so here I have 2,500 side effects, but after the filtering based on the side effect frequency, I think I got around 1,000 side effects to predict.
Oh, I have a suggestion for you.
Okay.
I would suggest that you go through them, and there has to be some sort of so-called ontology. I learned from this links that, okay, a lot of adverse reactions, actually the description overlap. And also another suggestion I have is, so you say allergic, and if you have involving immune system versus, so immune systems, you can have very different type of manifestation. So So what I'm thinking is that I would suggest, okay, maybe focus on the life-threatening one that people that could cause drug attrition, or at least cause the patient to discontinue a drug treatment. So, and then you would not have more so many adverse reactions, and a lot of them could overlap.
Oh, okay. But
you see what I mean? Because people use to use the same things, or in terms of severity-wise, you have different degree of severity. I would say then you perhaps will be able to have a better prediction, because you are predicting something. If you have, do you see what I mean? If you
I see. So do you have any suggestions for resources that like a list of severe side effects?
I typically look at the arrhythmia. I think Avi knows, and congestive heart failure. That a lot of terms are used for that. So you have cardiomyopathy, reduction in left ventricular reductions. So I will look into, there's NCI, there's a and actually, there's adverse reactions guide. I think you could read it. I cannot provide you the whole. Well, MDR Metro actually provides you, but then I would say don't use so many. Just focus on the most toxic one, the most serious one.
Okay.
Severe one. I would do that.
Okay, thanks for your suggestion.
Oh, by the way, I have a suggestion for you.
Yeah.
Okay. If you go and drug FDA and pull out the drug that has in the warning sections, okay, right now the label actually is very structured, and only in section 5 you have warning and precaution. And those are most, the most severe one that we list there. And also in the box warning, each label, on the top there's a warning. Those typically is life-threatening. So if you go through all the drug label, you will have a good idea also what constitutes a warning. And those actually are severe adverse reactions. And for the box warning that is life-threatening, then it's much rare though. But those are the good reference for you.
You asked me for the question, sir.
Okay, I guess that's for the questions. Thank you, Zichin, a lot. That was great. I hope
Thank you.
Everybody will attend next week, next webinar. Hope talk to you all later. Bye.
Bye.